The most common cancer in Taiwan is finally about to be conquered. Taiwan has highest rate of colorectal cancer. For years, it topped Taiwan's cancer rankings. Even Tu Qi Liang, our king of variety shows, couldn't escape its grip. But now we have new hope. Meet the Nanovaccine. Listen to how powerful this is. Not just colorectal cancer. You can freely swap out what's inside. Whether melanoma, brain cancer, pancreatic cancer, you name it. Cancers conventional immunotherapy can't touch. It can take them all down. Simply put, it's a truly personalized superweapon against cancer. I'm Dr. Zheng Dingjing, geriatric medicine. Could nanovaccines be the ultimate answer for all cancers? Do they have side effects? When will we actually get to use them? Let's use the key of science to unlock a long, healthy life. We already have chemo and targeted therapy. We even have immunotherapy now. So why is colorectal cancer still so hard to beat? The biggest reason early-stage colorectal cancer often has no symptoms. Nearly 70% of patients feel absolutely nothing at first. By the time something feels off, it's usually just incomplete bowel movements or frequent bloating. And though the government offers fecal occult blood tests, those can give false positives, like from hemorrhoids, which can easily also trigger a false positive result. So a colonoscopy is still needed to confirm. And if it's not caught early, late-stage treatment gets much trickier, far more expensive, and less effective. The second problem, the immunotherapy we have now, the newest and most hyped weapon in the cancer-fighting arsenal, only works for about 15% of colorectal cancer patients, leaving the vast majority of patients with limited treatment options. The other roughly 85% simply can't benefit from this kind of immunotherapy. Colorectal cancer has two types, microsatellite stable, MSS, and microsatellite instability, MSI. So what is a microsatellite? It has nothing to do with outer space or astronomy. It's just short, repeated sequences in our DNA. So microsatellite instability means the DNA itself is unstable. It mutates easily, so the tumor keeps releasing a large number of antigens. Think of it as a tumor constantly broadcasting abnormal signals. So our immune system can easily spot that something's wrong. Once it detects the abnormality, it launches an attack. With immunotherapy, these unique signals make for highly clear targets. That's why MSI tumors respond so well to checkpoint inhibitors. However, only about 10% to 15% of colorectal cancer patients have the so-called microsatellite instability, MSI type. The remaining nearly 90% of patients fall into the microsatellite stable, MSS type, meaning their cancer cells keep a low profile. They don't put out many abnormal signals for the immune system to pick up on, so immunotherapy simply can't find them. That's exactly why our traditional weapons have hit their limit. So scientists started looking for a new approach to go after the other 80 to 90% of colorectal cancers. That's when they turned to something entirely new that we're all familiar with now. Messenger RNA, mRNA. That's right. During the COVID-19 pandemic, we all got way too familiar with the mRNA vaccine. During the pandemic, it was developed at an unprecedented record speed and became a global game changer. That was the first time the public truly saw the power of mRNA technology. But here's the thing. The mRNA vaccine wasn't originally designed for the pandemic. Cancer was scientists' original target. The key is, it can teach our immune system. Past treatments, like chemo, basically just carpet bomb absolutely everything indiscriminately. Targeted drugs, on the other hand, surgically snipe at cancer-specific molecular weak spots. But mRNA is different. It doesn't fight on its own. It trains our immune army into a special force that recognizes the enemy. Here's how it works. It takes a piece of mRNA from cancer cells and delivers it to antigen-presenting cells, antigen-presenting cells, APCs. APCs are like intel officers. They piece together an ID of the enemy, like sketching a suspect for a wanted poster, then handing it to T cells, the executioners. T cells train on that sketch in advance until they know it cold. Targeted drugs, on the other hand, surgically snipe at cancer-specific molecular weak spots. That's why mRNA has become one of scientists' most promising new weapons. Sounds perfect, right? But the problem is, mRNA cancer vaccines still face two major clinical challenges in real-world human trials. The first hurdle is a delivery problem. mRNA is structurally very unstable. Enzymes in the body easily break it down, or innate immune sensors flag and block it. That triggers an interferon response that intercepts the message. Imagine you have a crucial suspect sketch, but before it reaches HQ, rain soaks it, wind tears it apart, or security mistakes it for junk and tosses it in the trash. So as a result, the message often fails to reach its destination. Second, poor presentation efficiency. Even if the mRNA gets successfully swallowed by APCs and translated into protein, when the proteasome chops it up, the cuts are mostly random and highly unpredictable in length. So very few fragments actually end up loaded onto MHC molecules for display. It's like a chef who's supposed to prepare 10 dishes to serve, but because the chopping is random and imprecise, only 1 or 2 plates actually make it to the table intact. So in short, the bottleneck for mRNA cancer vaccines is that the message can't get through, and even when it does, it's poorly displayed. So scientists started thinking, is there a way to make mRNA more stable and more precise? A great upgrade would be to give our mRNA a highly specialized transport package, essentially packing it up safely inside a protective gift box. That's a nanovaccine. Its core concept is to pack the message inside a nanoparticle. These particles are about the size of a virus, so our body treats them like one. Our APCs will then easily engulf it, avoiding the issue of the mRNA being selectively degraded or randomly discarded that we just discussed. First, the outer shell. So how exactly is this outer shell made? Using the fact that oil and water don't mix. With alternating layers of water and oil, we create a tiny layered oil droplet, like a millefeuille. It's called a lipid nanoparticle, or LNP. This nanocarrier is roughly the size of a virus. Just a few thousandths of a hair's width. That size lets it ride the lymphatic flow to the lymph nodes, and APCs can easily gobble it up. This solves the problem of mRNA never reaching APCs. Next, what goes inside? Scientists no longer toss in a whole strand of cancer cell mRNA. Instead, they use algorithms to pick the fragments easiest for the immune system to spot, the key pieces that spark the strongest response. These are called neoantigen peptides. In other words, it's like the chef chopping vegetables we talked about earlier. We pre-cut the ingredients, then pack only the best parts into the lunchbox. Once the APC takes it in, it can immediately load the right info onto MHC molecules. T cells recognize the enemy on the spot. That fixes the presentation problem we talked about. Nanovaccines can also bundle the antigen and adjuvant together. What's an adjuvant? Think of it as a seasoning that attracts T cells to the package. So packaging these two things together is what we call the co-delivery strategy. Immune cells get the instructions and an emergency alert at the same time. The result is a faster and stronger immune response. Now you might wonder, is all of this just wild science fiction or just theory? Actually, scientists have already made real progress. In 2024, a mouse tumor study in ACS Nano showed that scientists loaded neoantigen peptides plus 2 adjuvants into nanocarriers and found that tumor growth was significantly suppressed. Another U.S. team encoded the HIV-1 antigen, in other words, the AIDS virus antigen, into mRNA, then delivered it using lipid nanoparticles to get it into cells. As a result, he found that the induced antibodies were not only high in quantity, but also incredibly diverse in function. They could bind, block, and neutralize HIV. And after vaccination, they remained detectable for at least 41 weeks, broader and longer lasting than traditional protein vaccines. Now clinically, nanovaccines are still in early trials. The cancers they're most often tested on include melanoma, pancreatic cancer, and brain tumors, cancers that conventional immunotherapy is basically helpless against. But early results show they do activate T-cell responses, and safety is within an acceptable range. But the real excitement is the platform potential of nanovaccines. Picture this. A nanocarrier is like a universal lunchbox. The box stays the same, Just swap out the antigen fragments inside, and it can be quickly customized for different patients and cancer types. That means if the technology matures, every cancer patient in the future could get a vaccine tailor-made for them to reduce recurrence and even prevent metastasis. But of course, challenges remain. First and foremost, of course, is the cost. Designing and manufacturing a highly personalized vaccine for each individual patient costs far more than traditional treatments. Secondly, there's the issue of mass production and logistics. Nanovaccines must be prepared under strict conditions and rely on cold chain transport to stay stable. So even after the vaccine is made, getting it to the patient is itself a challenge. Thirdly, of course, it's effective only when there are significant antigen mutations in the cancer cells. This essentially means that cancer cells need to display unique surface markers that are clearly different from normal cells. As we mentioned, microsatellite stable colorectal cancer has fewer mutations, so it's still harder to identify. We still need more research and combination therapies to break through. But don't lose heart, because there are still many highly promising clinical studies currently underway and in the pipeline. In the global nanovaccine race, Taiwan's strengths lie precisely in the two most crucial areas, targeting and delivery. The team, led by academician Han Chung-woo at Academia Sinica, has long been dedicated to the development of drug delivery systems. They have designed a targeting peptide called PHCT74, which acts like a biological GPS installed on the outer layer of the nanoparticles. This allows the nanocarrier to accurately locate cancer cells within the highly complex environment of the human body. They've also developed targeted mRNA-LNP technology. This essentially gives the nano lunchbox self-driving capability, letting it find target cells on its own. Although nanovaccines for colorectal cancer are still in experimental stages, remember that colorectal cancer at stage 0 already has a near 100% cure rate. Even at stage 4, when it has spread, combining multiple treatments can still bring significant survival benefits and extend average survival by nearly 3 years. Perhaps in just 3 years, our nanovaccine will have developed to a much more mature stage. So to all cancer patients, don't be afraid. Work with your medical team to discuss your options and find the plan that works best for you. Nanovaccines are just beginning. What disease do you think will be transformed next? Cancer? AIDS? Or infectious diseases like the flu? Tell us in the comments. And don't forget to subscribe our channel. Join Team Health. Let's stay healthy together.